Hello everyone, my name is Shichang Xin and my advisor is Professor Guo Zhen. Here's my seminar presentation. The topic is bulk motion phase correction for optical coherence tomography and geography images. Okay, the outline consists of the first part introduction, the second part method and experiment result, and the third part discussion conclusion, and finally the future work. Okay, the introduction section. What is optical coherence tomography and geography, which is OCTA, is a non-invasive image technique that visualizes blood flow like in the retina and choroid, offering variable insights into various eye disease and vascular abnormalities with exceptional detail and precision. It provides high resolution that resolve images of retina and choroid vasculature. And why is there a motion in the images? These artifacts may arise from various factors such as physiological motion of the sample, including breathing or heartbeat, and external factors like vibration or instability during the imaging process. Even small movements such as minor shifts during retinal and cerebral vascular imaging can significantly distort collected data and impact the accuracy of vessel mapping. Okay, the motivation and purpose of this study. This study makes reference to the papers propose a histogram-based algorithm to extract and correct the bulk motion phase. This correction technique is crucial for eliminating motion artifacts that may affect the quality and accuracy of the angiography images, and ensuring reliable mapping of the blood flow in the retina and choroid vessels. And a way to eliminate bulk motion on this paper, implement a histogram analysis approach that works well for bulk motion artifact correction. When the tissue contributes to most of the pixels between two A lines in a big scan image. This will be explained in more detail in a later slideshow. Okay, here we reference two papers on phase correction. The first one is CCPGB. And here's the flow diagram of the complex correlated phase gradient variance. And the goal is also to achieve, compared to the phase resolved duplet variance, CCPGB can essentially eliminate the undesirable phase shift caused by bulk motion and trigger jitter, and it can be used to make microvessels. The difference with duplet variance imaging is the phase gradient. This equation is the axial gradient of the phase shift induced by red blood cells in the blood vessels. Okay, and this flow diagram is the approach of another paper on bulk motion phase correction, which focus on correcting the phase between two A lines by histogram-based analysis. So the first step, we convert the OCT data in complex form into the expression like A times exponential I times theta form. And then we divide the OCT data into intensity and phase terms. And this is the result of splitting the data into intensity and phase terms. The image data set I'm using here is an image of blood vessels in the mouses here. And the next step is to find the phase difference between any adjacent 2A lines, like J+1 A line compared to the J A line. Okay, this slide compares my result for finding a phase difference with the paper's result. And the left-hand plot shows the paper's result phase difference between adjacent A lines and plot of the highlight phase difference in figure B. And the plot on the right side is a result of my work on finding the phase difference. Okay, the next step is to analyze the histogram of the phase difference and find out the most distributed phase theta plot as the bulk motion phase between the two A lines and correct it on the J+1 A line. Okay, this slide compares the result of my histogram analysis with the paper. On the left is the paper's result, the result of the histogram analysis, and the result smoothed by the moving mean filter. And the graph on the right side displays the outcome of my histogram analysis and graph by applying a moving mean filter. And for example, we take 0.20944 as bulk motion phase between the two A lines and correct it on the J+1 A line. And finally, the last step is to correct the theta of the initial data from A times exponential I times theta by bulk motion phase theta plot, and then obtain the blood vessels imaged by OMAG method. And from the picture below, it can be found that the bulk motion phase correction method based on histogram analysis mentioned in the paper can remove the bulk motion quite effectively. Okay, and this is the experimental result of the bulk motion phase correction method based on histogram analysis that I refer to in this paper. We can find that the part of the bulk motion that is circled to represent the surface layer has been removed. And at the same time, the part of the blood flow signal that is pointed by the arrow has been retained. However, the effect of this method in removing the bulk motion is not significant in some B-scan images, and we can find that the part of the circle representing the superficial bulk motion is not eliminated, although the part of the blood flow signal in the arrow pointing to it is preserved. In addition, when we stack several B-scan images and observe them in the Z projection, we can find that after bulk motion phase correction, the horizontal strip noise in these two circles has been eliminated, but the small microvessels in these circles have been removed too. Okay, and then this is discussion and conclusion part. The discussion is this study introduces motion contributing factors, emphasizes the use of tissue map-based algorithms for correction, and compares the proposed method with other angiography techniques such as optical microangiography, which is OMAG. And the proposed angiographic algorithm CCPGB is phase sensitive but immune to phase genesis induced by bulk motion and trigger jitter, resulting in improved vascular visualization and superior vascular connectivity. And the conclusion is the proposed method effectively correct both motion artifacts using a histogram-based approach, and it enhancing the quality and reliability of vasculature imaging in the retina and choroid. Okay, and this is my future work. I will attempt to remove horizontal strip noise in the image and maybe try other smoothing filters on histograms to find the face. And last one, I'm continue to try to improve the method based on both motion phase correction. Okay, and here's my reference. That's my presentation. Thank you for your attention.